Hello guys, welcome to my YouTube channel. My name is Sibin and today I am going to explain you about isolation and purification of RNA. What is an RNA? Otherwise ribonucleic acid, which is an important biological macromolecule that is present in all biological cells. It is principally involved in the synthesis of proteins and carry out the messenger instruction from the DNA, which itself contains the genetic instructions required for the development and maintenance of the life. Now, let us discuss about the constitution of RNA in a typical cells. If we are taking 10 power minus 5 micrograms of RNA, it will have 80 to 85% of ribosomal RNA, which is rRNA, and 15 to 20% of small RNA, which is tRNA or small nuclear RNA, and about 1 to 5% will be mRNA. The problem with RNA. The RNA is chemically unstable. The spontaneous cleavage of phosphodiester bond via intermolecular transesterification. RNA is susceptible to nearly ubiquitous RNA degradation enzyme, which is RNase. The RNAs are released upon the cell lysis, and RNAs are present on the skin, and it is very difficult to inactivate because disulfide base conferring the stability and no requirement for divalent cation for activity. Sources of RNase contamination: ungloved hands, tips and tubes, water and buffers, lab surfaces, endogenous cellular RNases, RNA sample, plasmid preparation, RNA storage, chemical nucleases, and enzyme preparations are considered as the source of RNase contamination. The common sources of RNA contamination are contaminated through solutions and buffer and contaminated through equipments. When come to the contaminated solutions and buffer, we can use a good sterile technique and also treat solution with DEPC and make small benches of solution to avoid the contaminations of solutions and buffer. When come to the contaminated equipment, use pipettes, glassware and gelware with a sterile condition and bake glassware at 300 degrees Celsius for 4 hours. Use RNase-free pipette tips and treat equipments with DEPC. Now, let us discuss about the inhibitors of RNase. DEPC, otherwise diethyl pyrocarbonate, is mainly used for the inhibition of RNase. This will be also used for alkylating agents, modifying proteins and nucleic acids. First, we need to fill the glasswares with 0.1% DEPC and we need to let it stand overnight for the room temperature. For the solution, we may treat it with DEPC and add DEPC to 0.1% in concentration, and then we need to autoclave it. Then the DEPC will break down to carbon dioxide and ethanol. Now let's discuss about the material which is required for the RNA isolation. First of all, the substance from which RNA to be extracted. We can use any kind of substance like leaves, bacteria, virus, fungus for the isolation of RNA. Trizole, chloroform, isopropanol solution, TAE buffer and 70% ethanol. Procedure of RNA isolation. First step, we need to take 800 microliter of bacteria culture in a fresh append-off. We can use any kind of substance according to our needs. To this, we need to add 160 microliter of trizole, which is 1/5 of the culture volume. The solution was then mixed well by pipetting several times. To this, we need to add 32 microliter of chloroform, which is 1/5 of the volume of the trizole. Now we need to incubate this for 2 to 5 minutes and then we need to centrifuge it at 12,000 rpm for 15 minutes at 4 degrees Celsius. Now we need to transfer the aqueous phase into a new tube and add equal volume of the isopropanol. Now mix this well and centrifuge this at 10,000 rpm for 10 minutes at 4 degrees Celsius. Now discard the supernatant and thus suspend the pellet in 70% of ethanol. Again, centrifuge at 10,000 rpm for 10 minutes at a 4 degrees Celsius. Now discard the supernatant and air dry the pellet at 37 degrees Celsius for 10 to 15 minutes. Resuspend the pellet in 50 microliter of DE buffer and analyze the RNA sample quantitatively or qualitatively according to our need. Purifying RNA. To purify RNA, first of all, we need to break the cell and solubilize the compound in it. And now we need to inactivate the RNAs by the addition of guanidinium thiocyanate. This guanidinium thiocyanate is a very powerful denaturant. Now after this we need to extract the RNA using phenol or chloroform only at a low pH. Now we need to precipitate the RNA using ethanol or LiCl. Now stores the RNA in DEPC treated water at minus 80 degrees Celsius or formaldehyde, which is deionized, at minus 20 degrees Celsius. Selective capture of mRNA. This can be done by a method called oligo-DT cellulose method. For this, first oligo-DT will be linked to a cellulose matrix. Then the RNA is washed through this matrix at a very high salt concentration. Now non-polyadenylated RNA will be washed out. Then the poly-A RNA is removed under low salt concentration. Estimation of quality of RNA. The rRNA should form two sharp bands in ethidium bromide gel, but mRNA will not be visible. Use a radioactively labeled poly-DT in a pilot, then the northern hybridization should get a smear from 0.6 to 5 kV on the plot. Use a non-standard gene probe, for example GAPDH, in a mammalian cell. Then in the northern hybridization, there would be a sharp band with no degradation product. Quantitation of RNA. Nucleic acid will absorb ultraviolet light maximally at 260 nanometer. For a RNA, 1 OD is equal to 260 unit is equal to 40 micro by ml of ssRNA. The concentration in the sample is calculated by using the formula A260 into dilution into 40 is equal to RNA mu g by ml. Estimation of RNA purity by spectrophotometer. A260 to A280. Pure RNA will exhibit a A260 to A280 ratio within the range of 1.8 to 2. If the RNA exhibits a ratio which is lower than 1.7, which indicates the protein contamination in your sample. If A260 to 230, the purified RNA should exhibit an A260 to A230 ratio within the range of 1.8 to 2. If the RNA exhibits a ratio lower than 1.7, this indicates guanidine contamination in your sample. While the spectrophotometer reading do give an estimation of the purity of sample, they do not indicate RNA integrity. Why is RNA isolation so important? To accurately analyze RNA transcription, it is important to extract high-quality RNA from the cell or a tissue of interest with minimum RNA degradation. Keeping the samples according to our interest cooled for 4 degrees Celsius prior to RNA extraction will limit RNA's activity and safely preserves RNA for accurate analysis of gene transcription. Now let's discuss about the cons of RNA extraction. It is not very amenable to high throughput processing and difficulty to automate. Manual processing of sample can be laborious. Use of hazardous chemicals and chlorinated organic waste must be managed carefully. Starting with too much of sample or incomplete homogenization can seal off the membrane and or result in contamination with protein or genomic DNA. Incomplete cellular lysis can lead to low yield, and automation system for centrifugation or vacuum can be expensive or complex to set up. Application of isolation and purification of RNA. Clone genes, expression studies, Northern blotting, cDNA synthesis and gene production evaluation, and microarray are the applications of isolation and purification of RNA. Thank you for watching this video. If you have any queries related to isolation and purification of RNA, you can contact me via Gmail ID provided below. Thank you and have a nice day.